Okay, thank you for the invitation. This is really an exciting opportunity to talk to all of you. So I'm going to share my screen now.
Yeah.
And hopefully it will all work out smoothly. So I will be talking about, in addition to waveforms for TMS, I'll be talking generally about the topic of how can we push TMS further in terms of selective recruitment of neural targets in the brain. And I'll try to cover what I see as limitations as well as opportunities for this. Some disclosures. As an engineer working on technology, I try to collaborate with industry whenever possible and make this technology available to you, the users. So here are some of my disclosures. And of course, we're very grateful for the grant support we have received over the years related to these projects by the NIH, as well as other organizations. So first, some basics that I'm sure most of you are familiar with. Transcranial magnetic stimulation is a non-invasive form of brain stimulation that amounts to sending strong pulses of electrical current that can reach 7,000 amperes through an electromagnet coil that's placed on the subject's head. And this coil produces a magnetic field that in turn generates an electric field or induces an electric field in the brain. The magnetic field can be quite strong. It can reach a couple of Tesla on the surface of the coil. And then the induced electric field in the brain is strong enough to produce neurodepolarization. So it can make neurons fire. And then this firing can then produce various activity patterns in the brain and modulatory changes. And here I like to use the opportunity to contrast transcranial electric and transcranial magnetic stimulation. And you have heard some excellent talks about transcranial electric stimulation. So why do we do TMS? Well, let's compare transcranial electric and transcranial magnetic stimulation when we match the strength of stimulation of the brain. So let's say the strength that we apply is what's needed to get a finger twitch, right? Activation of the cortex. So when we match the stimulation that way, how do they stack against each other? Well, transcranial electrical stimulation takes about more than 10,000 times less energy than transcranial magnetic stimulation. So it's a lot more energy efficient. Also, if you care about that, it can penetrate deeper into the brain, even though it's non-focal when it does that. In contrast, transcranial magnetic stimulation has much less scalp pain or scalp sensation in general. And this is because the skull, which is not a good conductor, is transparent to the magnetic field. So to get the same electric field strength in the brain, we produce much less stimulation of the scalp with TMS compared to TES. And that really is the main reason why we use TMS. This is why we invest in this much more kind of expensive, large, bulky, power-hungry technology to get brain stimulation at and above threshold levels because of the tolerability. There are some other advantages of TMS. This insensitivity of the magnetic field to the skull and the scalp. It produces also kind of more, in a way, less variable targeting of the brain, targeting that's less sensitive to anatomical parameters. And however, we have a limitation of the depth of stimulation. And practically, you can push it to maybe a couple of centimeters from the surface of the brain into the brain. But even at that level, you get quite strong superficial stimulation. So basically, there is a stronger limitation of the depth of stimulation than with transcranial electric stimulation. And this is all to place TMS in the context of TS, which is the other very commonly used transcranial stimulation approach. Okay, so TMS obviously has some advantages and some limitations. Now let's talk about the limitations and how we can push them a bit further. So first, this is a slide that I like to show that I got from the internet, from the Mayo Foundation, Mayo Clinic. So they are generally excellent. And here they have an illustration of how TMS works, or is supposed to work. So you have the coil. There is what appears to be a magnetic beam coming out of the coil, and there's a target that is quite deep into the brain. And that this illustration gives the impression that that's how TMS works. Well, unfortunately, this is not the case. So in reality, the target is superficial because the field attenuates rapidly in depth. And this target can be characterized by some depth and spread. So in addition to that, the field is kind of spread out. So there's some effects of surrounding brain tissue. And within the target, there are different neural populations that get co-activated. Then there's transsynaptic activation of targets that are in the regions that are connected transsynaptically to the directly activated superficial cortical regions. Now, on top of this, we have these ancillary effects that are produced by the stimulation of the scalp, and as well as the auditory effect that TMS has. TMS coils, if you haven't seen one, it produces a loud clicking sound, and this loud clicking sound penetrates both through the skull as well as through the air, and the subject can hear it. And in fact, it can be a safety issue, so subjects wear hearing protection, like usually earplugs. And even with earplugs, you still have auditory activation, right? So effectively, you have this kind of spread of effects, both direct and indirect. And one of the goals of my career has been to try to see how many of these ancillary processes and sort of co-activations can we remove or attenuate so that you have the most selective activation of what you want, and therefore hopefully the most precise and targeted modulatory effects when you apply neuromodulatory paradigms like repetitive TMS. So how can one go about this? Well, in thinking about it, there are many things that can be done, right, in principle. So one can work on the spatial targeting, right? The more selective we're spatially with our induced electric field, the more selective we can be in the effects, in principle. And there are two elements there that can be worked on. There is optimization of the TMS coils that we use to try to make them most focal, as well as to go from really the external parameters we're applying to the internal parameters we're applying, which is the electric field. So this can be done with modeling, with realistic electric field modeling, which basically relates the TMS parameters at the level of the coil to what is actually induced in the brain. And I'm going to talk more about that. So another aspect is the temporal aspect of TMS, the temporal characteristics of the electric field. And there you have two key time scales. One is the parameters of the pulse, which impact how you recruit neural elements and different neural types. And another one is the temporal pattern of repetition of these pulses when one does neuromodulation with rTMS. And this links with higher level circuit dynamics and can produce various neuromodulatory effects, as we know. In addition to that, the neural connectivity, of course, matters, right? The brain is all interconnected. So actually, we can target deeper brain regions, even though we cannot stimulate them directly. And we can leverage for that anatomical, non-anatomical and functional connectivity between different brain regions, which can be imaged, for example, with DTI or resting state fMRI, or rather task-based MRI paradigms. There's the context of the brain. Now, there's a lot of interest in measuring the state of the brain, for example, with EEG and stimulating at the right time. and other aspects of brain context are important. And there are these ancillary stimulatory effects that we talked about, like the auditory stimulation or the scalp sensation that produce indirect activation. So working on all of these aspects can ultimately help us to improve the functional selectivity of TMS, and the field as a whole has been working on this in various forms. So I'm going to cover several of these in my talk here. I'll talk about spatial targeting, both in terms of coil optimization and using electric field-based dosing. I'll talk about the pulse parameters, and then finally, I will talk about the auditory effects of TMS and how we can reduce these. So starting with coil optimization, something that we have been looking at for a while is what is the relationship between the coil configuration, coil design, and the electric field characteristics in the brain. And some years ago, with my then graduate student ZD Deng, who is now at the NIH, we modeled what at the time were basically all the coil designs that you can see here in this nice gallery of some 60 coil designs. And we asked the question, how do they fare in terms of depth and focality of stimulation? And the conclusion was that there is a very clear trade-off between depth and focality. On this plot, you see how these coils are doing. Each dot, each circle with a number is a particular coil design. On the y-axis is the spread of the electric field on the cortex, which is the inverse of focality, right? The less spread, the more focal the coil is. And on the x-axis is the depth of stimulation. Again, a metric characterizing how rapidly the field attenuates in depth. So what you see here is that clearly the curve is diagonal. Note here, this is a logarithmic axis on the y-axis and the x-axis. So as the coils actually get larger, you get somewhat deeper stimulation, but significantly less focal stimulation. And the coils that are doing the best in terms of getting the best focality for a given depth are the so-called figure 8 coils, which is basically two circular loops of wire next to each other. And this has been the main workhorse of TMS coils for now some 3 decades. So what I have, at the time, I was like, well, okay, we are done with that. People have tried all kinds of designs and nobody has gone beyond this curve. So I thought that this is pretty much the limit. Then the past several years, I have had the opportunity to work with my brilliant postdoc, Luis Gomez, who has background in computational electromagnetics. And he was able to develop a very sophisticated computational approach that optimizes the design of coils. It actually synthesizes coils. Basically, we let the computer figure out what is the best coil to produce particular field characteristics in the brain. And essentially, the user has to specify what kind of surface, practical surface, you want to realize the coil on. We're not specifying the shape of the windings, the configuration. We're just telling it, okay, you want it flat or kind of curved or practical things like this. And then we give other specifications for what model to use, a simple spherical model or a more realistic model. And then the algorithm basically represents different possible current distributions on this surface and figures out which ones produce the closest to some desired specifications, like the target location, the direction at the target if that's specified, and some constraints like surface limit of the electric field on the surface and maximum coil energy. And for all of these specifications, it tries to optimize some cost function. In our case, it's trying to make the most focal stimulation for this particular depth and other constraints. And it turns around, and then in the end, it converts this current distribution on the surface that is specified to a pattern of windings. So in the end, we have a coil design. And this is an example here. So you can see that in the core of it actually is a figure of 8 coil. The current here, by the way, shows the direction of current flow in the winding. So blue is, let's say, clockwise, red is counterclockwise. So in the middle here, you have a figure of 8 coil, but then around it, there are these additional loops that increase the depth and increase the focality of stimulation. So it makes very much sense what the computer comes up with, but it has additional features on top of the conventional figure of 8 coil. So how far can this get us? Well, theoretically, if we don't have a limit on the energy and implementation of the coil, it seems that the fundamental limit is about a factor of 2, so a factor of 100% improvement in focality. Note that this is a logarithmic scale again, so this from here to here it's a factor of 2 increase in focality for a given depth. So, and that seems to be the fundamental physical limit of what can be done. The good news is that it is better than conventional figure of 8 coils. At the same time, it's a factor of 2 ideal improvement, right? And now the question is, what can we achieve practically? So here we can consider different planes, right, for implementing this. The most efficient one is if you just wrap the coil around the head like a helmet, and that gives you the most efficiency, and you can get the most focality, and everything is kind of very, very good about it, other than it's not very flexible, right? If you make a helmet, you can't really move it around for different targets. It may not fit on other people's heads and so on. So it's not very flexible. A flat coil, like conventional figure of 8 coils, is more flexible, but it's a bit less efficient. And it's kind of larger in size. So a trade-off between that is what we call a hat coil. And this was something that was this kind of surface, which looks like, like this, like a somewhat funny hat, right? Where you have this edge around it, right? So it's kind of illustrated, probably you can see it best here, kind of like a flying saucer or a symbol. And this kind of shape is actually derived by looking at many head models of different subjects and different positions on the head in an EEG coordinate system, and then you kind of spin it around to make sure that it can fit on most regions of interest over the head and on most people that you have considered. So it's sort of efficient, but still very generalizable. And that hat coil shape was proposed and pioneered by another postdoc in the lab, who was at the time working in Risto Moniemi's lab in Finland. This is Lari Koponen. And we have been using the same approach for our what we call FDTMS, focal deep TMS coils. So here you can see some examples that are implemented as a hat. And you can see another aspect of a trade-off here, which is, right, I said that we can get 2 times better focality if we have unlimited energy. Well, this shows you how much energy you need to get what improvement in focality. So in this plot, you have energy on the x-axis and spread, which quantifies focality on the y-axis. So you can see that with this kind of half sphere and sphere coils around the head, we can get the best improvement. Then with the flat coil, it's not as good, but we still get improvement over conventional coils. This is a conventional figure of 8, 70 millimeter figure of 8 coil here, right? So for all these, we're getting improvements. And with a hat coil or hat coils, because you can have many designs, we get this intermediate improvement between the flat coil and a helmet coil. So at the same time, it's very flexible. So it's a good trade-off. And the more energy you allow the coil to use, the more focal it can get. This is another important trade-off that we have quantified and is part of this optimization algorithm. So basically here we have we're able to improve the focality of stimulation depending on what energy we provide. Practically, for example, we can increase a bit the energy of conventional coils to get more focality, so we can get about 30% improvement for reasonable energy range, which is something. And it can have significant impact in, for example, mapping studies, or when you're trying to be very precise. Also, if you say, well, I'm fine with the focality of a conventional figure-of-8 coil, we can get much more energy efficient coils here, right? If we match the focality, look, we can have you need less than half of the energy of a conventional coil. So this can be very dramatic in terms of less energy requirements and less coil heating.
Angle, we've got a question here.
Yes, go ahead.
Is a hat coil convenient for stimulating structures outside of the motor cortex, i.e. Regions of different types of cells or different tissue / axon orientation?
Well, so they are, right, they're kind of two questions there, as I understood. One is, can we stimulate outside of motor cortex? And the answer is yes. And that's why we made this hat design so that you can place them on any of these points on an EEG system. I mean, anything reasonable, basically, in the upper half of the head, and you can stimulate there. That is, the coil will fit well. It will touch the head in the center, right? So that's why this is made to have this kind of general shape. In terms of recruitment, more selective recruitment of particular neural elements, right? That's something I'm going to talk about further. But basically, what we get with this is essentially, for the same depth characteristics of the coil as a conventional figure-of-8 coil, we get the field to be a bit more focused beam. And this is an example of this is what these things look like. I mean, this is an early prototype. Now we're making a kind of nicer one that we'll actually use in the human study. But we make them from a 3D printed former, and then we place the windings on the former. And then we use a little electric field measurement robot that characterizes the electric field of the actual coil. So this is, again, you get an idea of what this thing looks like. It is kind of like a wide helmet / hat. And here's some simulations and also measurements comparing conventional figure-of-8 with the hat coil. So you can see an illustration of this, about 30% improvement in focality. I put these circles here just to give you a reference frame, right? You see the yellow kind of area here, strong field is wider than here for the hat coil, and that is fairly significant. So we're basically now working on actual prototypes that can be used in humans, and we'll do a little mapping study after that to see how this design optimization translates into actual mapping accuracy. So here, I'll move on from this part, which is about how to make the coils basically more focal or deeper for a given focality, to now thinking about the characteristics of the pulse, the temporal characteristics of the coil pulse, right? So far we're talking about the spatial ones. Now let's talk about the temporal ones. And this is something that, as was mentioned in the introduction, I have been working on for a while. As an engineer myself, I have always realized that some of the limitations of TMS come from limitations of the technology. And over the years, we have developed a series of more flexible TMS devices, in particular the electrical current pulses that go to the coil. And we call this CTMS for controllable TMS. This is the evolution of it, and we have been fortunate to have partnership with Rogue Research, who have commercialized this device under CTMS or Elevate TMS name. And this has also made it accessible to other labs and I'm very glad to see the interesting studies coming from the labs of the likes of John Walto and Walter Paulos using CTMS now. So how does CTMS differ from conventional TMS? Well, it produces more rectangular electric field pulses. It's a bit more like an electrical stimulator, and you can produce various mono and biphasic and even multiphasic pulses. And you can control over some continuous range the pulse width. You can also control the directionality in a continuous fashion. You can go from more unidirectional electric field pulses like conventional monophasic pulses to more bidirectional electric field pulses like conventional biphasic pulses. And the majority of these pulses can be synthesized in pulse train that can be applied at relatively high frequencies, including these more unidirectional pulse shapes that traditionally cannot be applied at high frequencies with conventional monophasic TMS devices. To demonstrate what this kind of pulse control can do, some years ago, we ran a human study with this CTMS device, and the key person running the study was Stefan Götz, who was at the time a postdoc in the lab and is currently faculty at Duke. So there we tried an inhibitory or conventional inhibitory 1 Hz protocol in motor cortex. And we did a pretty standard design where we first had some probing pulses with a conventional TMS monophasic pulse to measure baseline excitability level in motor cortex. And then we applied 15 minutes of this 1 Hz protocol, which amounts to 1,000 pulses. And we used within subject 4 different pulse shapes, a conventional biphasic one, and then 3 other biphasic ones generated by CTMS that, as you can see, have these asymmetries. So 2 of them produce more unidirectional electric fields, pointing either posterior anterior in the motor cortex or anterior posterior, PA or AP. And one that was rectangular, but still kind of more bidirectional, like the conventional sinusoidal pulse. And at the end, we probed for excitability changes for 30 minutes. So these are the results. At the end of this 15-minute train, actually the CTMS rectangular pulses produced the most inhibition. And there were some significant differences between them, with the strongest inhibition produced by one of the monophasic pulses that was actually stimulating in the posterior anterior direction. Interestingly, the conventional sinusoidal pulse produced some inhibition, but that was not significant. And that links with some experimental studies that have shown variability or kind of unreliability of the inhibitory effect with conventional pulses. So the pulse effect clearly had, the pulse shape clearly had an effect on neuromodulation, even the time course of neuromodulation. You see with the biphasic rectangular one, there was this kind of actually increase in inhibition in the second 10-minute interval after stimulation that we analyzed. So the pulse shape does something. And one way to try to get a grip of what's going on is to see if there are any differences, as far as we can tell, in what these pulses stimulate. And this can be known from studies with conventional monophasic pulses, that the latency of the finger twitch that you get from TMS, the multi-volt potential, varies when you change the current direction for monophasic pulses. So similarly here, we had some differences on the order of a couple of milliseconds, which is on the order of a couple of synapses traveled through the brain, through brain circuits. So we have these kind of differences in latency between our pulses. And so in particular, the 2 unidirectional pulses had this large difference, and also the standard biphasic pulse had a pretty long latency as well. The longest latencies was the AP directed pulse, so going anterior posterior, and that is consistent with monophasic studies that showed the same effect. So basically, this means that we are stimulating some different neural population, basically, that has further longer latency to activate the corticospinal tract. And there are different interpretations here. It could be premotor cortex, right? Or some different part of the circuit in primary motor cortex. So this is really interesting. And as an engineer, I like to try to understand what is actually happening. So we have been running these computational models of the brain, where modeling the brain is very challenging, as you can guess. But breaking down our problems into simpler problems that are tractable with our current scientific and engineering abilities is, I think it's a great idea. Some reductionism to ask important questions like, what neurons do these different pulse shapes and pulse directions activate? So to get there, we have been working with several trainees, in particular Manna Bera, with contributions by Baosha Wang as well. Aman is a PhD student and in the lab, and Baosha is a postdoc, and that's in collaboration with Warren Grill from the Department of Biomedical Engineering at Duke. So we have been constructing, for these purposes, these head models that are just like, well, they are basically the models that are used for electric field modeling. But now we embed realistic representations of neuron morphology, so realistic neuron models, in the cortical layers of this head model. So we represent all possible 6 layers from layer 1 to layer 6. And this example of the cells, there are different kinds of cells that you can embed there. We generally have gone with the models that correspond to the lowest threshold cells, since they're most likely to get activated by TMS. And this has been published in a couple of papers now, most recently in brain stimulation. So let's see how does this process work. Well, first we have created, based on data from the Blue Brain project in Europe, we have created these very detailed models of human or humanized cortical neurons. So then we embed this, and we make such models for neurons representative of the different cortical layers. Then we embed them in our 3D brain model in the respective layers. And right now we have particular region of interest. For example, the vicinity of primary motor cortex that we populate. We're not populating the whole brain, but with big enough computer, you can populate the whole brain. Of course, you have to consider differences between the various brain regions. So then we do, we simulate the electric field induced in the brain. These methods have been around for a decade now. So then we get the local electric field distribution, and we impose this local electric field in our region of interest. What we illustrate here is the hand knob in primary motor cortex, where you have the representations of small finger muscles, for example. So there we impose this local electric field solution on our neuron models, and then we can convert these electric field maps into threshold maps. Basically, what is the threshold of the coil current to get activation of that particular part of that particular neuron placed at a particular location within a particular cortical layer. So it's really a very new take on these activation maps, which conventionally are just looking at the electric field strength, but now we can look at the response of the neural populations in different cortical layers. So one observation that is important here is that the one question that I have had and the field has had is, what is the key aspect of the electric field driving neurons? And there has been this hypothesis that it is the component normal to the cortical surface. And that's not unreasonable because the orientation of pyramidal neurons in layers 2, 3, and layers 5, which have the lowest threshold, are oriented normal to the surface mostly. But then you have all this branching around, and I have always been suspicious that that seems like too much of a simplification. And indeed, when we looked at how much the normal component, so in the second and third rows is the correlation of activation with the normal component of the electric field, that is the field normal to the cortical surface, there is a correlation, but it's not great. Now, if you look at the magnitude of the electric field, that is regardless of direction, just the norm, the strength of the electric field, now that really well predicts activation. So one conclusion from these models is that the electric field strength is a much more powerful determinant of neural activation in the cortex than the normal component of the electric field for TMS. And that has implications for interpretation of design and interpretation of studies. Now, going back to our question, how about this latency difference? Well, we can look at that, right? When we change the current direction, let's say posterior anterior versus anterior posterior induced current in the hand knob, which we give a slice here of the hand knob here, right? That's where it comes from. This is the hand knob, and this is basically a cross section of the gyrus of the hand knob. Right, when we change the current direction, the electric field distribution does not change at all. Just the polarity of it basically changes. So in our electric field models, we won't see anything. However, when we look at the neural activation, we do see a difference. And what happens in particular is that when you have posterior anterior pulses, you have more activation in the posterior wall of the precentral gyrus, where the hand knob is, right? This is the central sulcus here, and this is the precentral gyrus. So you have more activation here, and when you change the current direction, the activation shifts toward more anterior portions of these gyrus. And at the bottom is a difference map that highlights this difference. Basically, there's this shift of activation from here toward more anterior sides. So what does that mean? And how does that link with the experimental results? Well, here, when we look at this couple of pulses that are particularly unidirectional, when you simulate these, you see this effect, right? The one that's posterior anterior has more activation here, and then anterior posterior shifts the activation toward these more anterior sides of the gyrus. Well, again, so here you have the representation of primary motor cortex, and then toward the anterior portion of the gyrus, you have more of a premotor cortex. So there may be this shift of activation to more indirectly connected, that is, targets that are more indirectly connected to the corticospinal tract. And this is consistent with what has been found in monkey studies. This is work of Rathelot and Strick. And here they used actually mapping, electrical mapping in the cortex of a monkey. And they found that this is their basically hand knob. You can see here that you have, this is this more posterior part of the gyrus. And here you have direct representations of these muscles. Then when they looked at this for direct activation, then they looked at indirect activation. This is basically from tracing the cells that are directly connected to the corticospinal tract. And when you do electrical stimulation, you can see that there is a much wider spread of the sites that would activate the corticospinal tract electrically, right? And including this area here that is now more towards the crown of these gyrus. So basically all these dots here that you don't see here. So this is indirect activation. So we think that this is basically what's going on here. We're switching toward more indirect activation of the corticospinal tract, and these computational models with the embedded neurons appear to be predicting and explaining this quite well. So based on this understanding, it is compelling to rethink, at least it has been compelling to me to kind of rethink how we dose TMS. And this part is kind of somewhat speculative because we don't really have all the evidence we would like to have, but I can tell you what we have been doing. So the older I get, the more disillusioned I'm with the conventional approach of dosing TMS, which is you measure the individual motor threshold, that is the intensity that you finger twitch in some hand muscle over primary motor cortex, and then you move to some other brain area, let's say the dorsolateral prefrontal cortex for depression. And you use that intensity or something related to that intensity, like 120% of the resting motor threshold, to stimulate dorsolateral prefrontal cortex. So you go somewhere else, like in parietal cortex or wherever, and you stimulate relative to this motor threshold. So there are a lot of problems with that. I mean, first of all, the pros. Okay, well, it has been around for more than 3 decades. It's codified in TMS safety standards. If you tell people to move away from it, it's a big deal. And indeed, it may capture some generalizable head features and potentially some general kind of state of brain excitability. Right, so if you have a large or a small head or a thick or a thin skull, this may be correlated across different parts of the head, at least to some extent. And likewise, if there is some general kind of sort of excitability of the brain that is generalizable across brain sites, that may be helpful too, right, in terms of getting the right threshold. However, there are many cons. Well, of course, to do it well, first of all, you have to do this procedure of determining the motor threshold individually. Typically, this involves, to do it accurately, involves EMG. The motor threshold may be affected by the specific algorithm and accuracy of implementation of this algorithm and procedure. Then the local anatomy of the head is different in the primary motor cortex compared to the target where we want to stimulate, right? And that makes sense. The skull and scalp and the head is not the same in, let's say, DLPFC and motor cortex. Something that's well known is that the distance between the scalp surface and the cortex is different between different brain regions. And that's a huge factor for the field strength that you get in the brain. Basically, the coil to cortex distance is a huge determinant of how strong the field is in the brain. With the coil, when the coil is further away, then the field is weaker. And then when you think of how we get the motor threshold, we activate some representation, let's say, of the first dorsal interosseous muscle, like one finger muscle or another finger muscle. This is individual representation somewhere in the hand knob, and exactly where this representation is and what is the shape of the hand knob for a particular individual, and that varies individually, can have a huge impact on the motor threshold, but it has completely no relevance to your non-motor target. This representation does not tell you anything about your target outside of the motor cortex, and yet it affects this motor threshold. And of course, the specific excitability of the corticospinal tract may not be relevant to, let's say, DLPFC. Okay, so what's another way to go about this? Well, based on what I have been showing you from our simulation work, I become more and more convinced that if we have a good way of, assuming that we don't have a good functional readout, right, of the effect. I mean, if you can, let's say some people do false fiend threshold, right? If you're targeting primary visual cortex and you can get false fiends and you get the threshold, okay, that's a reasonable way to dose there. If you can measure some behavioral response or any electrophysiological response. TEPs, transcranial evoked potentials measured with EEG, is another promising approach, but they all have limitations. So what can we do beyond that for areas that are kind of silent, that we don't have some reasonable readout, functional readout? Well, based on our modeling work, it seems increasingly to me that targeting the actual electric field that gets to the, or dosing the electric field that gets to the target is a good approach to go, right? The electric field strength, as we showed, is a major determinant of neural activation, as one would expect. So how about we deliver the same electric field across subjects when we dose some region outside of motor cortex, right? I think that's quite reasonable. So we identify a target, and then we can optimize our coil position and orientation to get the strongest field at that target. And then we set the intensity of the field at this target to some specific strength that we want. And I'll talk more about this, right? That's a key question. What electric field strength do we want there? I call this EREF, or reference electric field. But let's assume that we have that value. Then we can use our computational electric field models. And also, we now have a very rapid way of optimizing the coil position and orientation. This is work developed by the postdocs in my lab, Moritz Denauer and Luis Gomez. Very exciting work. So you can do this optimization of the coil position orientation quite rapidly. One interesting point here is that to get these models to be pretty accurate, we have to measure the thickness of the hair, because we construct the electric field models based on MRIs, and there you don't measure the hair, but the hair can vary quite a lot. We have measured in our studies, we have this little gauge for measuring compressed hair thickness, and we have seen a range of 0 to 8 millimeters. And this can produce tens of % difference in the intensity that you need to get the same electric field, because of the, basically you get more spacing from the coil when you have thick hair. So we add the hair thickness to our models. So we distance the coil from the scalp surface by the amount of hair that's measured to make this accurate. How do you get this electric field reference? Well, there are various approaches. One can go from some modeling or other experimental numbers. Our approach has been to try to emulate actually existing paradigms that were based on the motor threshold. So we have taken a population from prior studies where we did electric field models, we did MRIs, we did electric field models, and we obtained motor thresholds. And then, let's say in a new paradigm, like one we have published recently in Brain Sciences, we're trying to target parietal cortex. And people in the literature have been targeting parietal cortex at 100% of resting motor threshold, right? So they say, well, my neuroscience collaborators would say, okay, let's go with that. And I'll say, well, but that's parietal cortex, so why should we use motor threshold? How about we use something better? But we want to still be kind of harmonious with this prior work that has used the 100% resting motor threshold. So I said, well, let's simulate that. We take this population of models and with motor thresholds and CAD models that we have, and we actually simulated the paradigm, what the paradigm would look like if we're delivering stimulation at the 100% of resting motor threshold over that target. And then we extract basically the average electric field strength that we get across subjects, right? That varies, right? There is a variation around this because different people have different motor thresholds and different head anatomies and all that. So we take the average electric field, and this becomes our reference field, right? So again, maybe I can illustrate that here. So we apply this then prospectively in the new study, and this is the correlation between the resting motor threshold in our new subjects and the pulse amplitude that was actually delivered based on electric field dosing. And there is a correlation that is poor, which is great, because it means that we're capturing a different kind of variability than explained by the motor threshold. I mean, there's a positive correlation, which is good, which means that, okay, there's some things that motor threshold is capturing, but we're capturing something else as well. So the mean is comparable. It's around 50% of maximum stimulator output. And that makes sense because we set the reference value to be the average corresponding to 100% of resting motor threshold. So we expect the mean to be the same, but the variation then is different. And interestingly, the variation for the electric field dosing was smaller than the motor threshold variation. Now, depending on how you look at this, could be good or bad, but I believe that it is good, that it shows that there are a lot more things contributing to motor threshold variation that are irrelevant to this particular parietal target. So basically, you can think of the two schemes as having essentially the same mean strength across subjects, but then different variation. Okay, so what are the pros of this approach? That is, we have applied it to a couple of studies, but basically it's still to be really validated. Well, it delivers the same electric field across subjects, and we know that D field is the main driver of neural activation in cortex. Then in terms of orientation of the coil, we also maximize the field and position orientation, and it happens that the best direction for strongest electric field tends to be aligned with this kind of normal stimulation of the cortex, which tends to produce strongest effects as well for neural activation. Then we have now this very rapid way of doing this optimization of coil position and orientation, and I invite you guys to participate in next week, organized by the University of Minnesota. Alex Opitz at University of Minnesota. It's on computational modeling and non-invasive brain stimulation. It's a symposium/workshop, and Luis Gomez is presenting on this method next week on May 28th. So check it out, Google it. It's a great workshop, a couple of days, a lot of nice presentations. Okay, so we have the kind of tools to do that. And going back to our method, you have comparable safety and tolerability because basically you get the same average intensity across subjects as with motor threshold based dosing, but you have this different individual variation in the pulse amplitude, which presumably captures the more relevant aspects of the local head and brain anatomy at the target. Now the cons, right? Well, it requires field modeling. Not everybody can do it, although now the tools are becoming increasingly available for those of you who have MRI included in your studies to really leverage that. Packages like SimNibs can actually do this right now. Of course, models have limitations, just like motor threshold determination has limitations. That's the case for models as well. We have good traction on some of them, not maybe as good as on others, but it's still work in progress. And already models are quite good. And some of this will be discussed at this symposium next week. And of course, we do not directly account for neural activation, which potentially could be integrated in some way in the models. But we have shown that the electric field is the strongest predictor of that. So going forward, we obviously need some validation. And I think one way to validate this is to look at whether we explain variability in outcomes better, or rather, if we reduce variability in outcomes when we do stimulation outside of motor cortex, when we leverage this approach compared to motor threshold approach. And that's not trivial because the two of them have the same average value across subjects. So you really have to look at the variability. Okay, so if anybody's interested in deploying this kind of approach in their studies and would like to talk to me, please reach out to us. We can guide you. And finally, I have about 5 minutes to just quickly tell you about our work on making TMS quieter. So briefly, the TMS sound is a big issue, and there's a list of why it's an issue. Besides the safety issue, it produces a lot of activation in the brain via auditory activation. So it makes the effective stimulation of the brain much less focal, and it's always synchronized with the pulse, which is awful. So we have been developing what we call quiet TMS, and it's a two-pronged approach. The first part of the approach is to make this pulse source that shapes the pulse to be much, much briefer, so that the nominant energy of the pulse is mostly beyond the human hearing limit of 20 kilohertz, right? We cannot hear above 20 kilohertz. In older people, it's even lower frequency. But if we push the dominant waveform frequency there, then we can hear less of the pulse. And also, it's easier to attenuate this higher frequency. Well, this is challenging because we have to make the pulse, when we make it 10 times briefer, you have to make it 10 times larger in amplitude. So we're working on that, making this pulse generator. And then the second prong of the approach is making a coil that's much quieter. And this work is currently led by Larry Koponen, the postdoc in the lab. And he has come up with this really ingenious approach of making a coil where there's an optimized coil, basically, and then you attach it to an external enclosure, and you attach it at the points where there's least mechanical vibration. So there's the least transfer of acoustic vibration to the surface of the coil. This is an example of a prototype coil that he has constructed. So here's what the performance looks like. And that's without the new pulse source with the brief pulse that we're developing. This is just the coil with a conventional pulse coming from a MagVenture device. So you can see here on this measurement, the peak sound pressure level, basically the peak sound strength versus stimulation strength. And this is calibrated relative to resting motor threshold, or average resting motor threshold. And you can see that our curve for the quiet coil is below all of the conventional coils that we have measured. And depending on which one you compare to, you have reductions of, in some cases, as much as 30 decibels. And for reference, 20 decibels is a factor of 10 attenuation in the waveform. And not only is it the quietest, but also, given how quiet it also is very electrically efficient. It produces some of the strongest. In fact, it's the strongest electric field output compared to other coils, apart from one which is a double cone coil. It's an angled coil. So for all flat figure of 8 coils, we're doing the best. So when you consider this combination of strong output and low sound, it really looks promising. And we hope that when we add these brief pulses, it will get even more dramatic. So that's all. In conclusion, I believe that the limits of TMS targeting are yet to be reached. There are certain physical limits, but they're yet to be reached. And I have tried to show you various ways in which we can make TMS more precise, more selective in terms of neural recruitment. And a lot of it incorporates new methods, like new technologies and new modeling approaches. So thank you very much. And I am certainly very grateful for the members of the lab and collaborators that I have had the pleasure of working with over these past years.
Brilliant. Thank you so much, Angle. That really was an amazing talk that I think myself and the webinar attendees found incredibly useful. So we've now got some time allocated for questions, if anyone would like to ask them now. And one thing I thought during the talk is that electric field modeling is actually available in BrainSight now, which makes it a lot more accessible. Here we go. Somebody's asked, have you thought about reducing coil vibration in QTMS as well?
Yes, that's precisely what this is doing, right? I mean, what is sound? Sound is vibration. It's mechanical vibration. When you send the TMS pulse to the coil, the electromagnetic forces inside the coil, and it vibrates. So there are a couple of ways in which you feel or sense this vibration. One is by the acoustic wave that it produces, right? So it produces wave in the acoustic wave in air, and also through your skull, and then your cochlea picks that up as sound. Also, the vibration, you can feel this through your scalp, right? Through basically pressure sensors in the scalp, and right. And when you make the coil quieter, you're reducing both of these. So we're the kind of we're getting close to running a human study with this quiet technology, and I am certainly curious to see how it will feel overall. And I would expect that the aspect of vibration of the scalp to be less as well, because again, we just make it vibrate less.
Thank you for that. There's another question. Is there a conflict between individualizing non-invasive brain stimulation and the need for standardized protocols in clinical applications for regulatory approval?
I don't think so. I think that individualization is generally appropriate when it makes a difference, right? Of course, you want to show that individualization is appropriate. Let's take, for example, a drug trial, right? You have to do your dose response characterization for a drug, and that may show that, okay, you don't need to individualize it, so you give the same dose to everybody, or you may need to individualize it to maintain efficacy and safety. And similarly with TMS, the assumption has been that you have to individualize. Certainly, if you want to get a finger twitch, you have to individualize the intensity and position of the coil, because otherwise you won't get the finger twitch, or you'll overstimulate. For other targets, I think that, from what I have seen, we have to individualize, especially when we do this kind of focal selective stimulation. When we do less focal paradigms, one can do analysis and basically see how much variation there is of for example, the electric field that gets to the brain. So in summary, my point is that when the response depends on the dose, right, and the response and the safety, then it makes sense to individualize it. If that's not the case, then there's no point of individualizing. So, and then that's the appropriate thing to do. And when it comes to regulatory clearance, well, you have to do the right thing to have your response and safety, efficacy and safety. So individualize when this is indeed has an impact.
Brilliant. Thank you for that. Another question's come in. Have you looked at pulse shapes that aren't rectangular?
Well, the pulse shapes that CTMS generates are sort of not perfectly rectangular. They have a bit of a kind of decaying tops. They're essentially portions of a longer sinusoid. We are very interested in studying more broadly the effect of pulse shapes, and CTMS does have some limitations, for example, in what pulses it can generate in a pulse train. It's much more flexible than a conventional device, but it has some limitations. So actually, with my colleague Stefan Gutz, we have been working for the past several years, and with another PhD student, Zhangxi Li, we have been working on the next generation of TMS devices. We call them modular pulse synthesizer, where it's literally like basically the sound card of your computer. You can synthesize any waveform at TMS power level. Of course, there's limitations. You can go crazy in terms of the energy, but you can cover the range in terms of energy of conventional TMS and maybe go beyond that. So with that device, we indeed should be able to more broadly explore the effect of pulse shape and optimize even the pulse shape itself in a less constrained way for particular aspects of energy efficiency and selective neural recruitment. So I'm very excited about this work. Again, we call it MPS TMS, Modular Pulse Synthesizer TMS.
Great stuff. Brilliant answer for that question. And I've got one more now. And can you please repeat the dosing of non-motor areas?
Okay, so I'll try to summarize that.
Yeah.
And well, maybe I should switch back, since I'm sure there was a lot of content for processing. I'll just go back to that slide so it is in front of us here. So it's actually conceptually quite simple. We just try to deliver the same electric field strength, induced electric field strength, at the cortical area of interest. right, at the target. So, for example, in this study that was published in Brain Sciences just literally within the past month, first author is Bainel, you can check it out. They were targeting parietal cortex. That's for working memory study. And we identified the functional target based on individual fMRI. You can identify it based on other anatomical landmarks, whatever. You basically define your cortical target. And then you say, okay, what coil position and orientation will maximize the field strength there? Or if you want, you can make it, the field go, let's say, normal to the gyral wall, so you can specify direction as well. And then we set the intensity of the current relative to maximum device output, or in DIDT, whichever way you control the current, right? With a big knob controlling pulse intensity or pulse amplitude on the device, you set it, and we do that in the model first, right? We set it so that the electric field strength matches a particular value. In our study, it was this magical numbers 56 volts per meter, which we got from what's explained here from a prior set of models where we basically simulated what the 100% of resting motor threshold would be on average. And on average, at this parietal target, it was giving 56 volts per meter. So I said, okay, well, we'll give the same average field, but now we'll give it exactly the same across everybody, right? And then you have some variability now in the intensity that is set at the device that comes from this model telling you to get the same electric field at the target, how you should set your intensity on the device. So basically, the computational model, and again, note that we try to do them as well as we can. We had these high resolution MRI scans, and we even measured the hair thickness to make sure that the coil in the model is positioned at the right distance from the scalp. When we put everything together, we got basically from the modeling, we got the parameters of where to place the coil and what intensity to set.
Brilliant. Thank you so much for that. We've actually had another question come in that's very much intimately related to that one, but I'll broadcast it anyway, just in case. Can you speak of more practical ways to decrease inter and intra-individual variability for output responses with TMS of the motor cortex?
I see the the implication that this is not a practical way. So I think that as complicated as this may seem to be, when, of course, yes, it involves getting an MRI scan, right? So clearly in cases when you won't have that, doing this approach is challenging, although one can think of then developing some predictors, for example, head size, and maybe other aspects of anatomy into some predictive model that tells you, well, roughly, set the intensity at a particular value. And then based on EG 1020 type of systems, you can localize roughly the anatomical target, right? So we can basically a combination of EG-based type, coil placement and some individualization of the intensity based on running a lot of models and developing some predictive aspect that can be easily measured. Like I'm giving, again, the head diameters example, although that may not be, sufficient. So that would be the kind of low-tech approach that would be an alternative to motor threshold-based dosing. I still think that when you actually in studies where you have an MRI scan, which is now the case in, well, a good number of research studies, I think it's really an opportunity to do this kind of dosing, and the tools that allow you to do this are increasingly available. And SIMNIBS is an example, and some of the work that we're doing, we hope that will be more available to users as well. Then I think that measuring basically the evoked response in the brain is, or some other readout is particularly promising, right? So if you have EEG, and you can measure transcranial volt potentials from your target, I think in principle that can be a very promising way to show direct engagement of your target. Of course, with TEPs, there are a lot of issues with noisiness. You have to clean up the noise. There's a lot of variability. However, if you set up the methods carefully and everything is done right, and there's still more research to be done in how to do this really online in a kind of closed loop fashion where you're titrating the threshold for a TEP, transcranial volt potential. I think that is, again, you need EEG for that. You need TMS compatible EEG, but that's a promising way because then you're directly measuring the brain response in non-motor areas. And of course, if you have any other way of sensing or quantifying the direct brain response, whether it is with a task or anything else that you can read out acutely in terms of response, that is probably the best way to show target engagement. So it the answer basically depends on what resources are available to you for a particular study or clinical work, which we are, of course, the research has to come before we're ready to deploy that clinically. But my hope is that more of this, and it is true that more of these methods are becoming, developed and hopefully will be available to the community to use as alternatives to multi-threshold-based dosing.
Brilliant. Thank you for that. I've got another question that says, would one need to characterize the output of individual machines to calibrate the output setting between 1 and 100% to the intensity used as an input to the model, which is DI over DT, or can we rely on some general numbers?
Yeah, so that's a good question, a good technical question. So for example, for modeling of the electric field, like SimNibs, it has models of various commercial coils, right? So you can generally you can find the model of your coil, and then you have to set the intensity as dIdt, that is the rate of change of the current. Some devices, in particular MagVenture, MagPro devices, they actually report this dIdt when you set, right, the most common setting for way to set the pulse amplitude in TMS devices is so-called percentage of maximum output. And that doesn't really say much, but what it is actually is the voltage on the capacitor, on the internal energy storage capacitor that powers the coil. So you can, and that's in volts. So you're basically charging this capacitor to different levels, and then there's a maximum voltage to which you can go. That's your maximum output. Then this translates when you discharge this capacitor into the coil, it gives you some rate of change, initial rate of change of the current. So the coil current starts to ramp up. So this is dIdt. So all devices have this percentage of maximum output, but that's really, it does not translate to anything that's particularly generalizable, because what you get out of the coil depends on the particular coil and the particular device, because the maximum stimulator output is different for different devices. So the dIdt is a bit closer to what's happening in the coil, and some devices, like, as I said, MagPro devices, report that. So in that case, you have this pretty straightforward relationship between dIdt and in the device and in the model. For devices where dIdt, that is the coil current rate of change, is not reported, one can, there are different ways to do calibration. One can simulate or measure the dIdt that corresponds for a particular coil, for a particular device, and how that corresponds to the setting relative to maximum output. And that's something where you may need to talk to the company that made the device or to engineers like us who can give you this. It's basically simple linear mapping. Again, for a given coil and a given device, there's a simple linear mapping between percentage of maximum output and the IDT. And then the DIDT goes in the model.
Brilliant. Thank you. So we've just got a couple more questions now, and I think we can start to end the webinar. Your new coil contains counterfield pairs. How could the combined coil consume less energy than that of a typical TMS coil?
It can, it does. So it's all about optimization. Basically, you have, right, yes, these counter fields, but what they do is they give you the right shape in a more efficient way than you could achieve with just with one pair of loops. And also, and I'm tempted here to quickly go back to the slides when I talk about this. So people can see what we're talking about. Okay, so here the coils in question. So the counter loops are where you have like red loops mostly on one side, and you have the blue loops. They're kind of countering that current flow. So what these loops do is they shape the superficial field, so it is basically more focal, and these larger loops on the sides contribute to deeper field. And then these coils in the middle, they're not perfectly circular. They're actually, the windings are more compressed toward the middle and more spread out away from the center of the coil. And this is a pattern, so-called kind of sort of asymmetric concentric pattern that has been shown before in the TMS literature, including the work of Larry Koponen, where he did some of the actually first computational optimization of TMS coils, and there was this pattern of more concentrated windings in the center and then more spread out windings toward the periphery. So this results in this energy savings, and yeah, it's all a matter of looking at the problem holistically and letting the computer figure it out rather than engineers who sit and come up with their own patterns. And fortunately, there is actually significant energy saving. And you can read about this in some of the papers of Larry Koponen and Risto Moniemi from 2015 and 2017 in brain stimulation, where they quantify these energy improvements from computational design and practical coils coming out of it.
Brilliant. Thank you so much for that. So this is the last question now, and it's about the practical combination of TMS and EEG. So for instance, if you want to stimulate the electric field in your target, your E-field model will predict you have the coil in a particular orientation and above a certain scalp location. But when you're doing TMS EEG, the pulse will cause muscle artifacts, which make the signal bad. And sometimes the orientation of the coil will be changed to reduce that muscle artifact, but it will produce a better EEG readout, even though the coil orientation is suboptimal in inverted commas. Can you comment on the compromise between the ideal configuration of stimulation based on the E-field and for a functional readout?
Yes, absolutely. This is a great example of how, the complexity of factors that we have to take into account when we're doing TMS dosing. And what I would do in such a case, yes, I would say, okay, this produces too much muscle stimulation. Okay, so we rotate the coil as much as we need to get away from that. We can even have to move the center of the coil to get away from particularly bad muscle activation. And then I would go back to my computational model, which can be run very quickly, enter the new position and orientation, and resimulate what is my electric field strength at the target, and then adjust the intensity of the coil current to get me the same electric field at the target. Now, the position won't be, orientation and position won't be optimal in the sense of most focal stimulation of that particular target anymore, but still we can make sure that we're getting the desired electric field at that target, the same electric field that we're intending initially, even though we have a new position and orientation. And that will be done by adjusting the strength of the current pulses, the intensity of the current pulses.
That's a fantastic answer. And thank you again for taking the time for such a clear response. That's it for the questions now. So I'd just like to take this as an opportunity to thank you again for your time, for all the pioneering stuff you've included in your talk.
Thank you. It was a pleasure.
So thank you so much.